My name is Clara. I'm a third-year PhD student. I work in the Genetic Functional Genomics and Biotechnologies Laboratory in Brest, in France. And my research focuses on gastric cancer, which remains one of the deadliest cancers worldwide. And in my project, I explore the concept of drug repurposing. The idea is to identify a FDA or EMA-approved drug that could be then redirected toward gastric cancer. And to do this, we screen over than 1,500 compounds using a 3D thread model derived from a human gastric cancer cell line. And we have identified a promising drug candidate. We are now trying to investigate its mechanism of action. So I use the Lumit Cell Proliferation Assay to measure the level of Ki-67 protein after treating the spheroid with the drug candidate. And Ki-67 is a well-established marker of cell proliferation, so quantifying it allows me to quickly see whether the drug has an impact on the proliferation of gastric cancer cells. There are several ways to measure cell proliferation, but most of them are time-consuming and they are also difficult to apply to 3D models. And before using the cell proliferation assay, I mainly rely on trypan blue cell counting and MTT assay. But both of these methods are less sensitive. They do not directly measure cell proliferation, and they can also introduce some variability, especially when working with spheroid, where a dissociation step may be needed. And the Lumit Cell Proliferation Assay allows me to measure a true marker of cell proliferation that really simplify my workflow. It works well with 3D cell cultures, require very few cells. The protocol is simple and reproducible. The sensitivity and the consistency of the luminescent signal allows me to detect even small changes in cell proliferation, so that helps me to make clear decisions and to move forward in my project. With the Lumit Cell Proliferation, we confirm this antiproliferative effect of our drug candidate, and we complemented this measurement with viability and apoptosis assay using the CellTiter Glow 3D and the Caspase Glow 37 assay. Our next steps will focus on better understanding the mechanism of action, looking more closely to cases cycle regulators, and testing and also additional cell models.